Okay, so this is the recording for the second part or part B of the integumentary system. Okay, so accessory structures. Again, when you identify these, you're going to have to state the tissue present in the accessory structure. So hair follicles and nails are both composed of stratified squamous epithelium. So same tissue as the epidermis. The hair follicle has a bed, bulb, lots of keratinocytes and melanocytes. Again, the amount of melanin is going to determine one of the factors that determines the color of the hair, as well as the type of melanin that is produced. Again, all of the hair shaft, hair root, hair bulb is all part of the hair follicle. The piloerector muscle is made out of smooth muscle tissue. So it's this column of smooth muscle tissue anchored to the hair follicle. When it contracts, it helps to elevate the hair because usually they lay up against the surface of the skin. And when they contract, they create a taller area and trap a pocket of heat to help warm up. This is why they tell you to layer your clothing in the winter. So in between each layer, you create a pocket of heat. Again, sometimes referred to as the erector pili muscle, same thing. Other accessory structures include all of the glands. You have the apocrine glands, merocrine glands, which are similar, and the sebaceous glands. So the apocrine and merocrine glands are a combination of stratified and simple cuboidal epithelia. The simple epithelia is typically in the duct leading away from the secreting cells, which is what we see here and we see here. On slides, it's not easy to tell the difference between the apocrine and the merocrine glands, but you can see the difference on the models. Apes are bigger than monkeys, so apocrine glands are bigger than merocrine glands. Also, apocrine glands are localized in certain parts of the body. Merocrine glands are merely spread out throughout the skin. Sebaceous glands are made out of what we call glandular epithelium. And that's shorter than writing out modified stratified squamous epithelium. So all of this is one sebaceous gland. It has different lobes. This would be one lobe. This would be another. You can also see a piece over here of another one. And another piece. This is a hair follicle, and this is a weird version of a hair follicle. So the sebaceous glands empty into the hair follicle. However, keep in mind that not all of them are going to look like they empty into the hair follicle. So in this picture, there are some items that you see that are not part of this system, but in order for them to do their jobs, they have to be present in the picture, their distribution. So the blood vessels, the red and the blue tubes, and the nerve endings, which is what we see here, anchored to that erector pili muscle. And then this black line anchored to the hair follicle. There would also be nerve endings going to, and again, this apocrine gland is not drawn to size. It should be much bigger. So yes, we can see blood vessels and nerves scattered throughout the body, but that doesn't mean they're part of that particular system. So 3 kinds of skin cancer. They're not the only ones, but they're the most numerous. Basal cell carcinoma begins in the stratum basal. So for each of them, you have to know their name, their full name, and where they start. So this is the lesion here, typically removed surgically. Squamous cell carcinoma is here. Notice it doesn't have the pigmentation of the stratum basal or the basal cell carcinoma. And that's because this is going to begin in the keratinocytes. And so it doesn't have the pigmentations. It's going to start closer to the surface. Malignant melanoma, so what we see here, and it starts in the melanocytes. This is the one related to exposure to sun. This is the one that can be life-threatening and can spread to other parts of the body. Next, we have the 3 different kinds of burns. And in some textbooks and in some rankings, there are 4. So, first-degree burns, and if he has a first-degree burn, it would be here. This damages the epidermis only. Second-degree burns damage the epidermis and the dermis. So the degree tells you how many regions of the skin are involved. Third-degree burns damage down into the hypodermis. Now with burns, you can also use the term partial thickness and full thickness. So partial thickness is typically the first and second degree burns. Full thickness burns would be the 3rd degree burns. With burns, it's not just the depth of the burn, but it's also the volume of tissue. So even though 2nd degree burns only involve the dermis and the dermis and epidermis, if it's over 50% of your body, then that can become life-threatening. The loss of the skin means you can't control water, you can't control temperature, you've lost, especially with a 3rd degree burn, you've lost sensory receptors. And so that can be life-threatening. In some cases, some textbooks, they also describe a fourth degree burn, and that's when you burn down all the way into the muscle and bone. So 3 degrees of burn. Okay, so for the drawings on this section, you're going to use the big box at the bottom. Now, sometimes you're going to have individuals. Like in this picture, you've got the Mirrocrine gland, and you've got a couple of pieces of Mirrocrine gland, which you can use. Here, you have a bunch of hair follicles, different versions. That white area is where the shaft has been removed. These have the intact hair follicles. This one has a sebaceous gland, which is all of this. This has a short piloerector muscle and a sebaceous gland. This has a hair follicle with a sebaceous gland next to it. This is an oblique version of a hair follicle. Here on the skin model, you can see the epidermis, dermis, hypodermis. You can see a mirrocrine gland. You can see an apocrine gland. Erector pili muscle. This purple is a sebaceous gland. This is what the sebaceous gland looks like from the inside. This is the outside of a hair follicle. This is what the hair follicle looks like when it's been cut open. Dermis, hypodermis. Another model. So here's again, an apocrine gland, mirrocrine gland, piloerector muscle. Different model. Hypodermis, dermis, epidermis, hair follicle, mirrocrine gland, apocrine gland, even though it's supposed to empty into the hair follicle, piloerector muscle, outside of a sebaceous gland, inside of a sebaceous gland. The whole apocrine gland, mirrocrine gland next to it, hair follicle, piloerector muscle, sebaceous gland, subcutaneous layer, dermis, epidermis. Another skin model, you have mirrocrine gland, epidermis, dermis, inside of a mirrocrine gland, hypodermis, hair follicle, apocrine gland, which is drawn correctly, piloerector muscle, sebaceous gland, apocrine gland, Mirroring gland, sebaceous gland, sebaceous gland, hair follicle from the outside, erector muscle, hypodermis, dermis, epidermis. And that marks the end of the part B of the integumentary system. Again, you're going to have to identify them. You need to do the drawings